Our next speaker, biomedical engineer James Collins. He's university professor and professor of biomedical engineering and co-director of the Center for Biodynamics at Boston University. He is, like so many of our guests this morning, heaped with awards, from Rhodes Scholar to McArthur Fellow and everything in between. It's raining Rhodes Scholars and McArthur Fellows around here. Dr. Collins' research focuses on developing nonlinear dynamical techniques and devices to characterize, improve, and mimic biological function. His specific interests include systems biology, that is, reverse engineering naturally occurring gene regulatory networks; synthetic biology, that is, modeling, designing, and constructing synthetic gene networks; and developing noise-based synthetic or sensory prosthetics. You may have heard about him in the news recently. Among his notable clinical accomplishments, he demonstrated that the dynamics of foot pressure distribution during quiet standing can provide a useful gauge of stiffness in patients like my mother with Parkinson's disease. His laboratory has also shown that introducing tiny random vibrations in foot pads markedly improves the ability of older people with peripheral nerve deficits to remain steady on their feet, which you may have seen in headlines only recently. Dr. Collins is a scientist who crosses the boundaries of engineering, mathematics, and biology to explore the complex mechanisms regulating biological systems. Join me in welcoming James Collins.
Thanks, Tom. What I want to do today is tell you a little bit about some of our very recent work in molecular biology, dealing with programmable cells. What Tom mentioned was some work that we are currently doing also on whole body dynamics, specifically dealing with vibrating insoles and enhancing balance control. What I want to give you, though, to start out is a sense of how does somebody like myself, I have a background in physics and biomedical engineering with a primary focus on whole body dynamics. How did I become interested in issues dealing with molecular biology and programmable cells? Well, it goes back a few years. If I could have the back slides killed. When I first joined Boston University, I was on the research faculty, which had the really nice benefit that I never had to attend any faculty meeting. Well, at our faculty meeting for our department decided to go for a very large grant from the Whitaker Foundation that would be multi-million dollars to establish a training program to train our students to go from genes to cells, to tissues, to organs, to whole bodies. The Whitaker Foundation likes to see kind of early efforts in bridging the gaps and or what was proposed. Well, it turns out our department made it to the final round and was going to be site visited. Charles Cantor, who was then the chair of our department, called me into his office and says, Jim, I'm a little embarrassed. We're going to be site visited. We have nobody bridging the gaps. I know that you're focusing on control theory applied to the whole body and studying how people maintain balance. Would you be interested in taking some of those notions, including some of your mathematical modeling, and bringing it down to the level of genes and cells? And he says, we have this cell that we've been engineering that would basically be designed to gobble up substance in the environment. Could you maybe model it and try to improve its control? So I thought about physics, so sure, we'll give it a shot. So Charles got all excited. He wrote down a bunch of equations. He said, I think these are the equations that would represent this system. We didn't know anything about molecular biology or cell biology, so I took the equations, and I'm leaving the office. Charles walks in and says, so Jim, stop for a second. You have 4 days to come up with something. The site visit's on Friday. So we go rushing back to my lab, and I got my team together. Again, people are working on how people maintain balance. We start playing around with these equations, and it turned out we didn't know enough to make adjustments, but the equations had nothing interesting to them. We couldn't make adjustments. They really didn't represent the system that Charles was trying to model. So Thursday night, I go back to Charles, and I said, Charles, I'm sorry, we have to punt. We didn't come up with anything. He says, no problem, Jim. I still want you to go out tomorrow and tell the foundation about what an engineer can do in molecular biology. So I said, okay. So the next morning, he wheels me out, and I give these half-baked notions about what an engineer could do. Well, we didn't get that grant. With good reason. Fortunately, my faculty colleagues did not hold it against us. What we did once we had more than 96 hours to think about this problem is, as engineers, we kind of sat back and said, okay, there's a lot of interesting things going on. Eric mentioned about the sequencing efforts and bringing together the parts list. In the late 90s, there was a lot of interest burgeoning about understanding the networks of these genes and proteins in the cell. And Eric talked about ways you can kind of crack open a cell and smear it on a chip and actually analyze what all the genes are doing. It's a relatively new technology that was very expensive and not as accessible in the late 90s. So whereas we first became interested in thinking about these natural networks, and I'll come back to this at the very end of my talk, we realized this was too challenging for us to make any inroads. So sitting back as engineers, we decided instead of trying to reverse engineer or pull out the structure of natural networks, that we as engineers could actually use our techniques and technologies, couple them like a biology, to design artificial circuits that we could get into cells to function according to some desired dynamics. And so we kind of sat back and said, okay, let's take inspiration then from electrical engineering and other branches of engineering to come up with cells with circuits with certain functionality that we could model firstly and then program them into DNA, encoding the circuits, and insert them into cells, bacteria, fungi, mammalian cells, and have them function the way we'd like to. So Tim Gardner, who was a very talented PhD student of mine, he and I kind of sat back and said, okay, this is a new area. Let's go after low-hanging fruit. So we thought about what would be a simple enough network that we could analyze and maybe get to work, but one that was complicated enough to have potentially interesting dynamics to warrant the effort, and that might also have some function from a biotechnology standpoint. So we came up with was this idea of a genetic toggle switch, which is motivated by digital toggle switches, the digital flip-flops, which lie at the heart of today's computers. What this simple schematic shows is kind of the general framework for this system. It's made up of two genes, called here repressor 1 and repressor 2, and two promoters. These are on switches for the genes. You design it so that the on switches are always on, and so each of the genes want to be on. You then set it up so that the protein that's produced by gene 1, shown here, wants to bind to the on switch for gene 2, shutting it down. And vice versa, you try to set up then the system so that the protein produced by gene 2 wants to bind to the on switch for gene 1, shutting it down. So each gene wants to be on, and each gene wants to shut the other gene off. So in principle, you should be able to design then a system, a switch, that will exist in one of two states. State 1, gene 1 is on, and gene 2 is off. And state 2, gene 2 is on, and gene 1 is off. In principle, you should be able to flip it, or flip-flop, between the two states by then delivering either a chemical or an environmental stimulus like a change in temperature that would, say, shut down the currently active gene. Say if you're in state 1, gene 1 is on, gene 2 is off. Deliver a chemical that will shut down gene 1 that will now allow gene 2 to come on. And once it comes on with sufficient strength that has produced enough protein, you can remove your chemical that is keeping gene 1 off because now gene 2 will keep gene 1 off. And now you flip the system from state 1 to state 2. Well, Tim Gardner, again, a very talented PhD student who had actually studied aerospace mechanical engineering at Princeton and designed helicopters, robot control helicopters, senior project. He and I started where we're most comfortable, and that was with mathematical modeling. Represented this system, did various analyses with pencil and paper and also on the computer, and basically showed that in principle this thing should work. Realizing that you're not going to get very far with mathematical modeling in biology, we began to go around talking to biologists who work on living cells and said, okay, what do you think? We'd walk into their office, we'd have the schematic, we'd have various mathematical results, computer results, and invariably the discussion would go as follows. What do you think? Oh, very interesting. Do you think it'll work? No. Well, why? Oh, they had various reasons. Things would get shot out of the cell. It would be very difficult to get this balance between the two. And invariably, the conversation would end as follows. They'd basically be showing us how they'll say, you know, guys, biology is really complicated. Well, somehow on the basis of basically this plot and our math results, we convinced Eric Eisenstadt, a very visionary program officer at the Office of Naval Research, to give us a grant for $500,000 to try to get this going in a real cell. The Office of Naval Research, like many programs, has a yearly gathering of their scientists. I was unable to go to it the year we first got support, and I sent Tim Gardner. Tim gave the presentation, basically including the schematic and our math results, computer results, no biology results, and he got slaughtered by the audience. The next day, Eric Eisenstadt called me on the phone and said, Jim, Tim didn't do so well. Nobody in the audience thinks this thing is going to work. I think it's going to work. I just don't think you two guys are ever going to get it to work. And he said, guys, I'm giving you a lot of money. Please go find a biotechnologist who can help you with this. And so we rushed up to Charles Cantor, a real cowboy in biotechnology. He was one of the early principal investigators in the Human Genome Project. And said, Charles, what do you think? He says, well, I think it's going to work for two reasons. One, it's a crazy enough idea that it should work. And two, you two guys are so naive about how hard it's going to be, I think you can get it to work. And with that ringing endorsement, he said, okay, here's some bench space in my lab. Again, we're studying people in my lab standing on vibrating platforms, vibrating insoles. We don't have stuff to do molecular biology. He led us into his lab and says, go to it. And what we did was set up a series of now genetic-based circuits in bacteria, in this case E. coli, that were based on the toggle. What I'm showing here is just a schematic. These are plasmids, rings of DNA, that are basically made up of biological components inspired by that earlier schematic. So what you can do in the microbiology lab are take chemicals that can act as scissors to chop out different parts, different promoters of different genes, and use chemicals that can act as glue to piece them back together to create rings of DNA with different components of a desired form. And so we have our two promoters, and we have here two genes of interest. This is a gene that produces a repressor that wants to bind to the promoter for this gene. This gene produces a repressing protein that wants to bind to this promoter, shutting it down. You can turn off this gene by delivering a chemical that's a sugar, and you can turn off this gene by heat shock, increasing the temperature. And what we do is we can monitor the state of the system, whether it's in state 1 or state 2, by also throwing in a third gene. This is a gene that produces a fluorescent protein that was found in jellyfish. So you basically say, okay, if you're in state 1, this gene on, then the cells will not be fluorescing. And if you're in state 2, this gene and this gene on, the cells should be fluorescing. So we get these plasmids into the cells. And what do we find? Well, we actually found that we could get, this is now real results from bacteria in the lab, that we could actually get this thing to flip-flop back and forth in a very stable fashion. So basically, you can start the cells in the off state, state 1, hit it with this chemical, the sugar. They all flip from the off to the on. You can remove that chemical, and they stay in the higher on state. There's a lot of variability in cells, so things pop around. But you can then flip the system from the high state down to the low state by delivering a heat shock, a change in temperature. Increase the temperature by 5 degrees Celsius. Boom. They now go from the on state to the off state. And these cells will stay there for as long as you want to look. The bacteria will divide every 20 minutes or so, 20, 25 minutes. We've looked out many, many days, and they'll stay in the off state if you start in the off state, and they'll stay in the on state if you keep on the on state. This kind of interchangeable notion of genetic circuits is very similar in electrical engineering. You can interchange parts. So we can take out the gene that's temperature sensitive and put in one that's chemical sensitive. This one actually gets turned off by an antibiotic, tetracycline. And we can produce then, instead of a chemical temperature toggle, a chemical-chemical toggle. Flip it with the sugar, keep it on, then you can flip it off by delivering the antibiotic. As an engineer, you'd want to characterize the different characteristics of your system, including switching time. So with our original chemical temperature toggle, what you do is you can basically start it in the off state, and we deliver the sugar, and it takes the system on the order of about 6 hours to flip. Pretty long time. Whereas if you started in the on state and now change temperature, it takes only on the order of about 20 minutes for the system to flip. We've since reduced this time down to about 20 minutes, but nonetheless pretty long. So what do we do with this first little project? Well, here's an ad from Accenture that came out in the early 90s, where they show fluorescing cells, bacteria tested as digital circuit. In many ways, that's what we did. We took a notion of a digital circuit, a flip-flop, and used it as inspiration to create a bacterial circuit, a genetic network, that was very similar to the flip-flop. It's a memory unit. It's actually a simple form of memory where the system can flip and remember some event to be, say, interrogated later. There is a lot of interest in this country and around the world in biocomputing, creating cells that do computing. I don't think anytime soon we're going to have jars of bacteria on our desks to surf the web for a lot of reasons, in particular the complexity, the issue of wiring cells, talk to each other through chemicals, not wires. But nonetheless, there is continued interest. In fact, one of the biggest developers and supporters of this is DARPA, the Defense Advanced Research Project Agency. When I first presented this work, it was at an amorphous computing workshop sponsored by DARPA. And as I'm going through these slides, the room is, yay, biological computing is here. We have a biological flip. Yay. Until I say, oh, wait a second, everybody. It takes 6 hours for this thing to flip. And the reason people were like, oh, gosh, what are we going to do with this, is that the switches you find inside computers take only fractions of seconds to flip. So imagine your little jar of bacteria, E. coli inside in your computer, taking hours now to upload the Boston Globe website. RCN will not help you out in that case. Well, we tend not to think of biological computers in the sense of doing computations in the way that your Dell computer does. Where I think that biocomputing has a strong future is in this idea of programming cells to perform different functions. And that is, can you use these circuits now as engineered devices to program a cell to behave in a certain way? And what we've become quite excited about is this idea of now taking our engineered networks, such as the switch, and interfacing it with natural networks. So bacteria, various organisms, have very complicated and sophisticated signaling pathways that can detect things in the environment, as well as different pathways that can have certain outputs or behaviors on the basis of some detection. So we thought, wouldn't it be neat if we could take our circuits, say, for example, our switch, and couple it now to natural pathways that could sense something in the environment, as well as have some readout of some interest. And so what we've done as kind of an easy scheme first was to show, could you create a programmable cell that could act as a sensor for something in the environment? That is, wouldn't it be nice if you could have a dish of cells that you could place in the environment that could sense a bad chemical, a pathogen, that you could interrogate to see, was there a detection event? And it turns out that the toggle switch, which has memory, it'll flip and stay flipped, gives you this really nice ability. So what we did was basically take the toggle switch and couple it to a natural pathway in bacteria that senses DNA damage. So it's called the SOS pathway, save our ship, that once the bacteria's DNA is damaged, this pathway gets activated, and we'll try to repair the DNA. We coupled our switch to it so that when that pathway gets activated, the switch flips. So we can start the switch in the low state, and it'll stay stable for as long as we want to look. And as you deliver increasing amounts of some DNA-damaging agent, which might be mitomycin C, which is a chemical used in chemotherapy, you basically can detect remarkably small levels of mitomycin C, where now you go from the off state to the on state. And as you increase the concentrations of your DNA-damaging agent, your cells flip more and more until at very high concentrations, they all flip over. Similarly, you can use it with UV irradiation. So ultraviolet radiation will damage DNA, our DNA, as well as bacterial DNA. And we were able to show that these little sensors now that are inside, they're basically based on whole cells, can flip in the presence of increasing amounts of UV radiation. We thought we'd get clever and see could we make a more complicated system. And what we got intrigued about was, could we design a crowd sensor? So could you design a system that could harness or sense the density of cells? So there's a lot of talk about communication in the conference. Turns out bacteria can communicate with each other. They actually will produce very small molecules, small chemicals, that will pass in and out of the cells that they use to get a sense of how many other bacteria are around them. And what we were able to do was say, okay, can we harness that pathway now to basically create a crowd sensor, a density sensor, that, say, could execute a function such as once the cells got to a certain density, too many in a dish, they start killing themselves. Or, say, once they get to a certain density, they start producing a protein. And so what we're able to do is basically, again, now take our toggle switch and now couple it directly to this intercell signaling system, this bacterial communication system, that was able to actually now flip in the presence of increasing density. So the cell is off, the switch is off, and as you increase your cell density, you actually have more and more cells flipping to the on state. Turns out this has biotech applications in that there are a number of companies that are using cells to produce proteins of interest. That what they need to do is allow the cells to grow up to a certain density, and then they whack them with chemicals to produce the protein of interest. But it turns out that it's incredibly expensive to whack them with those chemicals. And so what we've been able to do is show that you can actually program cells themselves to flip on and produce the protein once they get to that critical density. What we've very recently gotten excited about, kind of in the spirit of Mark Klepner's talk, is now moving from cells, could we actually engineer viruses to become even cleverer invaders? So just as there are viruses that want to go after us and other animals, there are also viruses that go after bacteria, called bacteriophage. They'll get into a bacteria, make a bunch of copies of themselves, and then they're really good. They got their exit strategy. They'll burst open the cell after they made multiple copies, having harnessed the DNA machinery of the cell, to basically now go and invade a bunch of other cells. It turns out one of the big challenges in modern medicine regarding infectious disease is dealing with chronic infections. Underlying chronic infections is this notion of biofilms. Biofilms are communities of bacteria that basically exist. They get encased in a matrix of polysaccharides and attached to a surface. Most bacteria exist in biofilms. They don't exist floating around in a free swimming state. And it turns out that 60% of chronic infections involve biofilms. They're very nasty. They basically are very difficult to eradicate because bacteria in biofilms are highly resistant to antibiotics by a factor of about 1,000 compared to free-floating bacteria. Well, we kind of sat back and said, okay, that's a tough problem. What could we do? Could we somehow, as engineers now, take advantage of the fact that these little viruses that kind of look like lunar modules that can invade bacteria, could we engineer them to make them even better invaders? So inspired by Keanu Reeves in The Matrix, we basically designed now viruses that could enter and destroy the biofilm matrix as follows. We basically redesigned these guys so that they would express on their surface. Mark showed how different organisms will change their coating. What we designed is so that they would express on their surface an active enzyme that would break up and degrade the matrix of the biofilms, exposing the cells for then invasion by the virus, which then would multiply, propagate, produce a whole bunch more of these viruses that could then go to the next layer. So you, by layer, have these engineered viruses go after the bacteria. Well, we actually found we could do this beautifully. We basically start with very high amounts of biofilm, in this case, and when we actually introduce our engineered enzymatic phage, we can actually get it down to levels where it's basically no different from what you'd find if you had no biofilm. So what can you use these things for? Well, this will be my next to last slide. Tim Gardner and I first got motivated in this space by being partly encouraged by some then success in gene and cell therapy. Presently, gene therapy is not a very popular area to be working in, but it's going to, I think, become popular again as additional techniques come into play. And in particular, cell therapy is going to increase in interest, whether it's from stem cells or other cells-based schemes. The big challenge in each of these, first in gene therapy, is can you develop an effective technique to get your therapeutic gene of interest into enough cells without harming your patient? In the case of cell therapy, it's can you actually encase the cells appropriately so that you also don't harm the patient? Once we get around those challenges, the next big challenge will be can you actually control the genes of interest in a safe and meaningful way? Turns out there are already available gene switches, separate from the toggle switch, that you can buy from companies. Each of these switches are kind of like a pressure switch. That is, if you, say, had it as an equivalent of a light switch, in order to keep the light on, you'd have to keep your finger on the switch. And that is in the genetic switch, you always have to have a drug present or a chemical present in order to keep it on. In the case of the toggle switch, it's actually different. It's like a typical light switch. You come in with just a transient scheme. You can flip it on, and it will stay on without you having to keep your finger there. So the idea is the genetic toggle switch, which was just now extended to mammalian cells by a group in Switzerland just this week, is that instead of having to always take drugs, perhaps the person would only have to take a drug once to flip the cell or the gene on, and then perhaps even more importantly, if the person is not responding well to that gene, take a pill once to flip it off and keep it off. Well, interesting in this regard, when Tim Gardner and I first came up with this notion, we went around and talked to a number of pharmaceutical companies and said, hey, guys, what do you think? Wouldn't that be great? And they looked at us, God, what do you guys think? What is your business model? We sell pills. It's a true story. We said, oh, but, patient compliance, I don't have to take it once, safety. It's like, oh, no, guys. Next. Well, there are various schemes in terms of biosensors, already mentioned out in the field, biotechnology. The idea of, again, you can program the cells for producing proteins of interest. Biocomputing, again, kind of coupled back to whole cell biosensors. And really less Flash Gordon, and what is really happening now is this idea of functional genomics. So this challenge we have of trying to figure out the function of the different genes that Eric talked about, one of the real powerful tools using transgenic animals. Well, you'll take an animal, you'll knock out the gene, and see what happened to the functioning of the animal. Well, it turns out that many such genes, when you knock them out, remove them at the embryonic stage, the animal doesn't develop well, or perhaps dies. So the idea is you could use something like a toggle switch to have the gene of interest under the control of the switch, allow the gene to be expressed as the animal develops, then you can knock it out, knock it down, so that then you see what happens when it's shut off, and then knock it back in. And then knock it back out, you can have a reversible system because the switch is reversible. Second, and more relevant to some of the things that Doug was talking as well as Eric, is this idea of gene expression studies. We're now actually using these probes as means to go in to uncover the structure of natural networks. We're looking in bacterial systems, trying to gain insight into why are some bacteria really good at resisting antibiotics. We're also moving into mammalian cells, trying to understand things such as the nitric oxide pathway, as well as various protein networks underlying HIV. So I'll leave you with this slide, is that appropriate for this conference. Born to network, is networking in your genes? Well, it was not in mine up until about the late 90s when Charles Cantor, with a strange offer, pulled me into the space. But it's a marvelous time as an engineer to be in molecular biology, to begin to think about the workings of the cell and how engineering principles can give us new insights into how the cells are functioning that potentially could be translated into medical advances. Thank you.
That's wonderful. Thank you. Thank you. James Collins, thank you so much. I don't know about the rest of you. I feel better already knowing that there are focused, great minds like this, bringing new antibiotics and all the rest of it here. Thinking about programmable cells, a genetic toggle switch, genetic-based circuits. Jim said each gene wants to be on. Each gene wants to shut the other gene off. Who knew? Humankind is already a giant biocomputer, right? Fabulous. It's great. Also, I love the insights into the parry and thrust of research grant battle and the research itself. This is a real insight for those of us who are not in that world. Just tremendous. And programmable viruses. I mean, the mind boggles again and again this morning, but it also listens and learns.